binding of other proteins that we call transcription factors that can also help RNA polymerase bind to the promoter. So in essence, here's the promoter region on our DNA. Here's our RNA polymerase. There's a couple of ways that we can help the RNA polymerase bind to the promoter. The first one is our sigma factor. And as you remember, our sigma factor binds to the Tata box. And that's at minus 35 and minus 10. And then we may have a couple of other helping hands, some other transcription factors, that may also help RNA polymerase bind to the promoter. And having all this coordinate regulation regulates how well RNA polymerase binds to the promoter. Yeah?
I thought the Tata box was part of the eukaryote.
Well, it's the minus 10 and the minus 10. The minus 10 and minus 35 in bacteria. It's the same thing as the Tata box. They're the same thing. It's where the binding of the RNA polymerase occurs. So in eukaryotes, this sigma factor is like 23,000 different molecules. It's a lot more complex. There's transcription factor 2H and 2D and 2 blah, blah, blah. Simplified in bacteria. But their region is the minus 35 and the minus 10. So the protein's affinity for binding to the DNA and bringing RNA polymerase will help turn up the amount of transcription that occurs. Okay? So both using sigma factors and other transcription factors allows us to regulate which proteins are turned on when. So here's the minus 35 region, here's the minus 10 region. Those are the two regions of the binding of the sigma factors. Okay? So the sigma factor will bind there. Here's the start site of transcription where we make messenger RNA. There also is potential upstream elements that also can help the RNA polymerase bind. We'll just call them upstream elements. But they're sequences of DNA that also will help the RNA bind. These regions here, the minus 35 and minus 10, are found in all bacterial genes. The sequences will vary from species to species, but for example, the E. coli promoters all have the same minus 10 and minus 35 region. All the streptococcus ones have the same minus 35 and minus 10 region. This is a very similar sequence in all genes. And again, this is binding for the sigma factors. So most E. coli promoters recognize sigma 70. This is the one that I said is that housekeeping genes. Those are everything, life is good, we just got to keep our cell alive genes. Those are sigma 70s. But that sigma unit, sigma 70, can be replaced by one of 6 different sigma units. For example, sigma 32 is the one I said is the heat shock one. We just threw our bacteria in boiling water, we need to save ourselves. So sigma 70 will be replaced by sigma 32, and this will turn on a different set of genes from our housekeeping genes. Because, well, the bacteria are about to die, so they're going to save themselves. Okay? So on the RNA polymerase, the sigma 70 is replaced by sigma 32. Sigma 32 now recognizes a different set of promoters, and these are promoters for keeping us alive when we're dying in boiling water. That includes chaperones that help keep proteins folded in the correct formation, even if we're in boiling water. But those are the type of genes that are turned on by the heat shock sigma factor. Yeah?
Now, those just attached to the
RNA polymerase. Mm-hmm.
Like, but where do they come from? They just get released how do they?
That is a very complicated process. Yes, in essence, they get released when the cell recognizes that they're in a heat shock type environment, when their proteins start denaturing, things like that. There's people that actually have spent their entire career studying these heat shock proteins. But yes, it's, in essence, things start denaturing, the sigma 32 gets expressed, it replaces sigma 70 on the RNA polymerase, so that now it can turn on genes responsible for not dying in boiling water. Yes?
So because there's chaperones on the proteins, they don't denature in heat?
Yes. They only have a finite capacity though. at some point, if you heat the bacteria up hot enough, even the chaperones won't work. But they're trying their best to keep their proteins in the correct conformation. But
So
obviously,
that's
if you boil bacteria for like an hour or so, you'll kill them.
That sigma factor still allows the transcription process to happen.
Yeah, so the sigma factor is responsible for bringing the RNA polymerase to minus 35 and minus 10. Sigma 70 brings it to the minus 35 and minus 10 of your everyday housekeeping genes. When sigma 70 is replaced by sigma 32, now it stops turning on the everyday genes and it turns on we're dying in boiling water heat shock genes. By that replacement, we go from doing the housekeeping genes to the I'm dying in boiling water genes. So by replacing that, it's able to coordinate expression of all those different genes at one time. And bacteria have this set that we know so far. So we have ones that are important for stationary phase. Stationary phase is when the bacteria they don't have any more nutrients left, they're running out of food and they just gotta hang about. The ones for swimming, sigma 28, that turns on all the flagella genes and allows bacteria to swim towards food. This would be something, whatever reason, we're in a liquid media, we need to get towards food, those will go on. There's one for sporulation, I don't know if it has it on this list. One for nitrogen levels, yeah, I don't remember what the sporulation was. I should remember, the lab across the street from us used to work on that, but I don't remember exactly what it is. But they have a different set of subunits depending on what conditions that they're in. So bacteria that produce spores have one that's specifically saying, hey, we're in a bad environment, let's go produce a spore, and it'll turn on the spore genes.